All right, welcome to OmegaMatters. I'm Kristina, this is Bill, and today we're talking to one of the giants in the omega-3 research field, Susan Carlson. We'll talk to her about a recent publication, what she sees coming next in the field, and how to use her research clinically. So Susan, welcome to OmegaMatters, and thank you for talking to us today. And you and my dad have quite a history, so you guys can fill us in on that a little bit.
Yes, well, I mean, the way I think about our history is we changed institutions. We went across the state line at sort of the same time, and we've done a meeting together. But I think it was a joke for years because even though we worked a mile apart, I mainly saw Bill in the Amsterdam airport or various places like that. So
that's right.
I think there was an era where we were all on the road a lot, and
Yeah.
so that's kind of the joke that I always think about. I didn't ever see Bill in Kansas City.
Well, right, and then the gap between early human development and atherosclerosis is pretty big.
Mm-hmm.
So
Yes.
we were both interested in fatty acids, but in very different ends of life, and so we didn't have as much together. I always feel bad when I think back on our 2008 conference and how you and I had this great idea to propose this fall conference in Kansas City, and we convinced the board to do it, and all gung-ho, ready to go, and then I left town.
I think you even told me right after the vote. That's my recollection, that very day, right after the vote, you said, I'm moving.
Well, that would have been.
It all worked out because I made you work anyway.
You made me work. It worked out wonderfully.
And I liked your lecture yesterday because you've pulled them back together. You're talking about premature birth and premature death. So maybe we'll talk about premature birth today.
Exactly. Okay,
Yeah.
Tina.
Yeah, so I will, other than their personal connection being from Kansas City and both interested in omega-3s and fatty acids, I'll provide some background on Susan. She's been in the area of omega-3 and omega-6 fatty acids in the field of early life health for both mom and baby. The majority of her career, a PhD in nutrition, and currently the A.J. Rice Professor of Nutrition in the KU Department of Dietetics and Nutrition at KU Medical Center, and was just finished a term as the Associate Dean of Research in the KU School of Health Professions. And she's been at the KU Medical Center for over 20 years and really focused on fatty acids like DHA and arachidonic acid and their effects on brain development in infants and preventing preterm birth. So we highly recommend looking into her PubMed citations if you want to see the depth and breadth of the work. It's very impactful. And we're very excited to have her here today. So,
and let me also congratulate her for being elected a fellow of the American Society of Nutrition. Just
yes,
inducted last week, right?
So you were a fellow last year, right, Bill? But you were on the call yesterday.
Yeah, I was last year. Yeah, but you were long overdue. I was shocked when I got in that you were not in the list.
Well, there we go.
Anyway, we corrected that problem.
That's good. Good. Well, so today we wanted to talk about your most recent paper. It's published in EClinicalMedicine, which is the online journal for Lancet, I believe. And the title is Higher Dose Docosahexaenoic Acid, or DHA, Supplementation During Pregnancy and Early Preterm Birth, a randomized, double-blind, adaptive design superiority trial. So a lot of scientific titles have a lot in there, and this is no different. Let's just start with the basic question you were asking in this study. What was your study about?
So really our basic question is 200 milligrams, which is becoming a standard dose in most prenatals that women take in the United States, is that adequate, or is 1,000 milligrams superior to reduce birth before 34 weeks? As you know, birth before 34 weeks are the babies who stay in the hospital longer. They're more subject to a lot of problems like retinopathy of prematurity, BPD, necrotizing enterocolitis. There's a whole bunch of conditions that are really common. The late preterm babies, those are the ones that are born after 34 weeks, they tend to stay maybe a week and go home to mom, and generally in the United States, they have a pretty good outcome.
Mm-hmm. Yeah.
So that was our primary question.
Right. The earlier they come out, the babies have less and less time to develop, and there's more that we have to do outside
Yes, and
But
I'm
the-
glad we chose a superiority trial because a placebo trial we did not think would work in the United States. In the end, we found out that half the women in our study were taking DHA when we enrolled them. So I think that they probably would not have wanted to stop taking DHA, and I would have felt it wasn't that ethical
Mhm.
to tell them no. So we just gave everyone 200 milligrams and said, stop taking whatever you're taking now. We will guarantee you get the standard prenatal dose.
So that's,
Yeah.
that's the superiority word in the title.
Exactly.
And adaptive was the other word in there too. What does that mean?
So adaptive design is considered the ethical way to do clinical trials now, and it's becoming more and more common. And what it means is you assign more subjects to the winning team, so to speak. So when you have two arms like we did, and the reason it's more ethical is more women get assigned to what is probably the best outcome.
Mm.
And it's also more efficient because you can finish the trial more quickly if you get a p-value that you decide on what probability you're going to accept at the front end. But if the probability had been 0.99, we would have stopped the trial after 850 women. We didn't achieve that, of course, but that's
Okay, so the assignment of women to treatment or placebo changes over time because somebody's looking in the background at how the outcomes. Is that how that works?
Yes, so they're obviously in a blinded trial, you have to be careful about how you do this.
Mhm.
So the way this trial was designed, after 300 women were equally assigned, 150 in each group, then every 3 months, there was a determination about whether one side was doing better than the other, and a new randomization schedule appended onto the last one.
Uh-huh.
So that was done by people in the biostatistics department who really had no other involvement in the trial. So none of the PIs or anybody working on the trial knew.
So you didn't know as the ratio changed, you weren't aware of it.
You didn't know which, you knew the ratio was changing, but you didn't know which group was which, right?
Got it.
Yeah, so you
Interesting.
ended
It could have
up,
been the 200 milligram group that was winning, right?
I know, it's happened.
Yeah.
and so at the end of the day, 576 women got 1,000 milligrams and 524 got 200 milligrams.
Yes.
For basically the second half of half of their pregnancy, essentially. They were recruited.
Yes, at least they had to be no more pregnant than 20 weeks gestation, but as early as 12. I
Mhm.
think the average was right around 4 months.
Yeah.
Pregnant.
Yeah, so that's what's interesting about the fatty acid studies is often they are in the second half of pregnancy versus like the folate and those kind of studies are more the actions at the beginning of pregnancy. So it's always thought that was kind of nice that you could intervene in midway in pregnancy and still start to see effects. So that's the basis of the study. Let's go into some of the results and then we can talk a little bit more about how you guys did the statistics, because you've just really pushed the boundaries on a lot of these statistical methods and we want to make sure we understand them. And it's good. It's good that we're progressing in that way. So the main results of the paper, do you want to just quickly go over the main results?
Yes, so the main result was a 2.4% early preterm birth rate in the 200 milligram group and a 1.7% in the 1000 milligram group.
Mm-hmm.
But in the meantime, which had a probability of 0.81, posterior probability that the 1000 milligram was better. But obviously that's not the same as a probability of 0.99.
Mm-hmm.
In the meantime, the Australians published the ORIP trial and they published a secondary analysis of that trial, which showed that the women with low status had an advantage that was statistically significant, but women with high status didn't really have any advantage.
Mm-hmm.
So one of the things you're allowed to do in a trial, if the trial is ongoing, you can change your protocol and based on new information that's come out. So we changed our protocol to say that we would do a subgroup analysis based on what would be low or high as per the ORIP definition. And that's still kind of it's considered a secondary analysis. It's not the primary analysis is the one I told you, but the secondary analysis you were allowed to do and publish with your primary analysis. And what we found was that women who started with a DHA status in that low range had a dramatically lower difference in early preterm birth. It went from 4.1% in the 200 milligram group down to 2% in the 1000 milligram group. And the interesting thing was, and why it wasn't more different in the group as a whole, is that the 53% or so of women who were above that status, it didn't matter which dose they got. They had a very low rate of early preterm birth.
So it was about a 50-50 split above and below this
No, we
target.
didn't do a median split or anything like that. We picked a number that, interestingly, in our KUDOS trial, which was done between 2006 and 2010 before prenatals were on the market, 90% of the women were below 6% at baseline, and 10% were above. And to me, it's just, I mean, I think we knew that the prenatals had come into the market, but we had no way of knowing how penetrant it would be in the sample we were looking at. And it's also, I think in some ways strengthened the study, because before we got this dramatic effect of supplementation, even with 600 milligrams. And I think that's partly because everybody was so deficient. In this particular trial, you could say half of the women were not particularly deficient in DHA at the time we enrolled them. I mean, many of them were not, because they had been taking DHA since they got pregnant, perhaps.
Yeah.
We tried to catch them with their prenatal visit, which is a little bit later.
Yeah.
And were you also, I'm just remembering a previous conversation, I think, where you were saying that your population that you recruited for your study, that whole population actually had a lower rate of preterm or early preterm birth than the general hospital population. Is that right?
That is true. Yes, the preterm birth rate in our hospital, I can't tell you off the top of my head, but it's in the 4 or 5% range. So 2.4% is very low. Now, remember, there was nobody that was getting a placebo here. There may have been some women who weren't taking their capsules, but everybody was given either 200 or 1000 milligrams. So in the paper, we talk about the fact that, you know, we can't say that 200 milligrams is beneficial, but the fact that we had such a low rate of early preterm birth in that group, and also it's much lower than the US national average. For preterm births, which is somewhere around 3.5% for early preterm births. So 2.4% is considerably below that. And you could infer from that that 200 milligrams was doing something. But of course, we can't prove that based on the way the study was designed.
Yeah, could also be volunteers or different people than just the regular community patient, right? People who volunteer to be in studies.
I think it's quite interesting, because we had a full range of people from high school education or last to college professors in the trial. And I think we did a pretty good job of capturing a pretty generalizable population when we considered all 3 sites together.
What were the 3 sites? KU and?
Cincinnati, Ohio, and Columbus, Ohio. But even within cities, there are differences. And I was just talking about this morning to someone. In the 3 sites we had, the Columbus population was a low SES hospital population. The Cincinnati was. The Columbus was a pretty high income clinic. And then we had Kansas City, which was from our hospital, which is a broad range of people with different backgrounds. And all 3 sites, women started with a different baseline status. So the Cincinnati group started lower. They were also less compliant based on their final version. So again, whenever you're dealing with a low SES population, you get that kind of result. The Columbus, Ohio population had higher DHA status at baseline than ours, and they finished higher too. So they were very compliant. So I think I think it's something people don't, probably if we'd gone to a different site in Cincinnati where we were capturing the same kind of women as in Columbus, we would have had much different outcomes. And even Kathleen Gustafson, she was conducting the Panda trial at the same time ADORA was going on. Her mom started higher and ended much higher than our moms. So clearly taking it at the beginning and also very compliant.
Yeah. Makes a difference. I really, about these results, I also like how in the paper you do the number needed to treat analyses. So you say for the women who started out in the lower omega-3 status, you would need to treat with 1,000 milligrams, 43 women to prevent one early preterm birth. Is that how that's read essentially?
Yes, that's correct. And I think that puts in I think it was obstetrician who was one of the reviewers who asked us to put that in. It wasn't our plan, but that reviewer wanted that number. And I think that's the way people who are practical in taking care of patients think, like, okay, if I recommend this to all my patients, how effective would it be? And you know, to prevent one for treating 43 is actually pretty good.
Yeah,
very good.
That's very good, especially for a nutrient that has, it's relatively safe. There are definitely some things we're looking at with higher and higher levels, but relatively safe and cheap to get. And then the number for the women who started off with a high DHA level, for them, you would have to treat 500 women at the
Exactly,
higher
which means it's probably not worth it, right?
Right.
Give them the 200 milligrams. The one thing I will add to this is that, and you may have missed this in the paper as a whole, is that in the secondary analysis, there was a pretty dramatic reduction in preterm birth with the higher dose. It was quite over 3% difference in preterm birth, and we don't want to minimize late preterm birth. It's got its own risks. It's just not as serious a situation. But in the developing world, babies who are born preterm, that's the major cause of infant mortality because they don't have NICUs and all the support system that we have in the Western medicine.
Yeah.
Did you see a prolongation of gestational age? I mean, were they?
We did not. And I think this was, well, I'm sorry, maybe we did by one day. I wasn't focused on that because we found that so many times. It's the prolongation of gestation, though, is actually related to this reduction in preterm birth. We showed that a long time ago in a study where we combined the domino data from Australia and our kudos data. And that was the revelation that it's probably not that we're prolonging gestation as much as we're preventing early preterm births and preterm births, which lend to a smaller baby. So
and you found some other benefits as well for the omega for the 1,000 milligram versus 200, didn't you, on other endpoints as secondary endpoints?
Well, we looked at safety, and that was one of the things you always do in a trial like this. And again, the posterior probabilities for the 1,000 milligram dose being safer for mom were in the 0.8 to 0.89 kind of range. But for the babies, there were several outcomes that were significant. You know, they had a probability much better than that, even that there was less risk to the infant in the higher dose group.
And I don't know if we were talking about this before we got on, and I don't think we've fully come back around to it, but you keep saying posterior probability, and I don't think we've defined that one yet. Yes. Loosely defined, it's kind of the cousin to a p-value. It's trying to say the same thing as when we otherwise would use a p-value. So can you? Yes.
So I mean, we've all used p-values for all of our scientific career, right? But the biostatisticians they're both probabilities. They're both talking about probabilities, but they're determined in a different way. So the p-value is conditioned on the null hypothesis, that is the chances you're not going to find an effect. And the Bayesian posterior probability conditions on the observed data from the experiment. And the way I understand it is, and I was reading some of these things yesterday, the Bayesian people, the way they talk about it is the Bayesian results allow you to fit into the whole context of that research area, whereas a p-value is kind of unique to that particular study. You're studying some outcome, but okay, you find in this study, but it doesn't really tell you whether that study is an outlier or, you know,
Yeah.
it doesn't really help you get to the answer to your question, which is a health question, as well as the posterior probability question.
And it's more, is it kind of thinking there's an 80% chance that this is the 1,000 was better than the 200 is how you would maybe say it in layman's terms, and makes statisticians cringe.
Yes, yes. I mean, you can, just like I tried to explain earlier, I mean, in this study, 0.81, you could say, well, probably the higher dose is better, but it does allow you to probe into it and see who's really benefiting, which is what we did. And what we found is probably you don't need to take the 1,000 milligram dose if you're already been taking DHA for a while. So
yeah,
the more data you can get, the better it helps people understand kind of where they might be able to make a decision about
and
what they want to take.
I think in nutrition, it's also important to it's just not as black and white as maybe some other sciences where there are omega-3s in everyone's body all the time, no matter what. And it's not like a drug where if you're not taking the drug, it's not in your body. So there's always this spectrum that you're then intervening on. And now we hear there's a baseline creep in the US where levels for pregnant women are likely increasing, at least in certain populations, which is great, but that's going to affect every study that's trying to look at this, unless they look at baseline blood levels, and then you can take it into account. But just having that little bit more nuanced view statistically versus it's significant or it's not. It was an effect.
Exactly.
I think that's
exactly. And I think that's a concern with a lot of nutrition trials, because as people begin to study a nutrient, they have an impact on people's behavior. And we can't forget that there are industry out there who listens more closely than physicians. I mean, they're on top of any new thing that's coming out, and because that affects their bottom line. And so I don't think they started putting I mean, there's no evidence that 200 milligrams is good for pregnancy. It's just, we said, okay, let's make that number, right? And that was one of the reasons we did the trial, frankly.
Yeah.
So now there is evidence.
Yeah, right. Yeah. The 200 story, I mean, so you still recommend 200, and that's just a kind of a general, just a good idea without solid evidence base? Or?
Yeah, well, I mean, it's better than zero now that we know that some work, probably some works, but obviously that would be the next trial that one would do, is find women who are low and then do the trial with a placebo or 200 or 1,000. You design the trial.
Are there other big trials going on right now? Do you know?
I'm not aware of any, but I'm not sure that I would be actually. I haven't looked on NIH lately. I've been so busy. I haven't looked to see what other trials might be funded out there around this question. But as you know, the Cochrane review that came out in 2018 said we don't need to do any more trials.
That's right. That's right.
And
quite a statement.
Yeah, that's what it says. But unfortunately, it didn't really tell women how much DHA they needed to take, right?
Right.
Well, I understand the dose used in those 70 some trials in the Middleton Cochrane review was something in the neighborhood of 1,600 milligrams a day.
I don't think that's true. I think they did a kind of a secondary analysis, if you would call it that, from the Cochrane results, and came up with the idea that it's probably over 500 before you get an effect on early preterm birth.
Yeah, I missed that.
Yeah, because they included all kinds of randomized trials in there with low dose, high dose.
Right.
Yeah, it was a big.
Right.
I remember looking at that. So this does lead a little bit into, we can either talk about kind of the comparison to the other big study, which was AURA, that you've kind of alluded to, and how that affected how you did your statistics, and perhaps maybe why you saw an effect and they might not have. And then the other thing we wanted to talk about was potentially like the blood levels and the standardization and being able to use those to be able to understand these studies better.
Yeah.
So we can go either way. Let's maybe start with ORAP since we're kind of on randomized controlled trials.
Well, I guess I don't know anything officially that would allow me to say why we got different results from women who started with higher baseline DHA.
Mhm.
As you know, they have published a paper saying that there is a relative risk of getting a high dose if you start with a high baseline DHA status. All I can say is that we've published this paper and we addressed that issue for our study. And we had in our highest quartile of baseline DHA status, we had one early preterm birth out of 262 women, which is somewhere around 0.4%, I think.
Yeah.
So in our trial, we don't see that happening. Now, what we do see is if we look at the quartiles, we see kind of a flat amount of early preterm birth across every quartile in the high gross, where it drops from 4.5% down to like, basically we didn't have any early preterm births in the highest quartile of 200 milligrams.
Yeah.
So I think that we do agree on one thing, and that is that women with low DHA status have a lower early preterm birth rate when you give them a high dose.
Yeah.
Compared to 200 or a placebo.
Yeah.
The other part will have to be resolved, but I would not feel any compunction to not give 1,000 milligrams to women regardless of their baseline status, because as I said before, we reduced preterm birth.
Mhm.
So when I was talking to the people who were doing their work in Zimbabwe, where of course the people don't even have a good source of DHA that they can afford, it's probably very safe in those conditions to give everybody a high dose, and it would probably be wise. You'd probably get a better result. Birth.
And
but if people wanted to be very cautious, then they should test, which I think is what you're leading into here.
Yeah, right.
Yeah. We like testing, but it begs everything becoming more personalized and giving, blanket recommendations. Your study just showed it, is that depending on your blood level, your personal nutritional status, it did affect what you kind of needed, which I think makes sense. And so, especially in like the US and Australia and countries that have more like healthcare based pregnancies, the omega-3 story is it's similar to, it could be similar to folic acid supplementation in that it's generally recommended, but again, we have this dose issue. And then what if we were able to test pregnant women for omega-3 status at their 12-week visit and then be able to give a higher or lower recommendation for omega-3s, or even, like you said, we could just do a standard higher dose potentially, but this would be
more motivated. I think you're more motivated if you know you have you're at risk.
Right, I agree with that. I agree with that.
What do you think obstetricians are in this?
So you, Bill, I've been thinking about this because there's 4 million deliveries, give or take, in the US every year.
Okay.
And our friends in South Australia are getting the blood from everybody in South Australia and analyzing it and making recommendations based on that. I guess I'm a little more pragmatic. I would like to find a better way. So one of the things we did was we asked women using a 7-question survey about foods that have DHA. And what we found is that women at baseline who were taking less than 100 milligrams, they had a much lower early preterm birth rate if they got the 1,000 milligram dose. So based on that, that could be a pragmatic approach. So we went to the OBs in our unit here, and they decided to administer a red cap survey that has those 7 questions. And of course, the one thing we didn't know was whether women could fill it out on their own reliably. So we're just finishing that experiment, but we have like 90 women who filled it out and then agreed to have my research associate interview them the way she did during the study, right? Because the question is, can they fill it out on their own? And what I can say is that I've seen that maybe the first 75, and there were 2 women who were outliers. One of them was low on the thing and she was high when she interviewed her. It turned out that when she took the questionnaire, she thought, oh my gosh, I should be taking DHA, and went out and bought the supplement. So I think it's going to be predicting almost perfectly who is below 100 milligrams, which would then, so we're about to do another small trial that's in discussion with DSM to do a compliance study where we actually get blood too. Because as you know, there's not a complete correlation between blood DHA levels and the intake level. And so I think a low intake probably represents a low blood for the most part, but there are probably some women who are low intake who have more DHA. I don't know. I mean, we don't know that, or there'd be a perfect correlation if that were the case. So we'd like to have both the questionnaire. What we're going to do is, if women have a low intake, then we're going to randomize them, but we're going to get blood and send it up to you guys and get the blood spot analysis too, to see how that goes.
And for the pregnancy story, it seems like it's more of a threshold effect at a lower omega-3 status than we usually talk about for, optimum health for adults in general. And when you're getting up to the higher statuses, people will say, oh, I take a supplement or I eat fish occasionally. And then they think they have they're an 8% omega-3 index. And we know that almost always is not the case. Yeah. But when you're talking about pregnancy, the levels we're talking about are much lower than what we're talking about for the optimum. It's on our scale, it's more of a 5%. It kind of matches your 6%, a 4 or 5%. And so when someone does take a low dose omega-3 supplement, or just like a normal supplement would be 300 EP and DHA, that might be enough to get them above that threshold. It's more maybe a deficiency kind of status situation than optimizing.
But there's optimal for preventing preterm or reducing risk for preterm birth, and then there's way more outcomes in a pregnancy than just that, right? And
right. There's a lot
higher
of
levels are going to be.
There's a lot of interest in. There's just an RFA that came out today that I first saw today anyway. That's about cardiovascular health of pregnant women and strategies to, what is pregnancy doing to them long term, and a higher dose may well be better in that particular case.
Yeah. Well, even some of your other outcomes, you said that were benefits, 1,000 milligrams on some baby-based, infant-based outcomes. That,
yes, that's true. In our last trial, I think I may have mentioned this to you, that we had a placebo or 600 milligrams. The boys whose mothers got 600 milligrams have a completely different inhibitory response. The girls, it didn't matter if their mothers got it or not, but there was a sexual dimorphism on benefits. So the boys benefited from getting it, whereas the girls
were already
perfect, fine without it.
Right, exactly.
So again, that's like brain, and your mind goes to things like boys and hyperactivity and ADHD.
Sure. Yeah. Right.
So it was really on inhibition, really, their ability to, how their brain is seeing things and how they're reacting. But cognitively, all the behavioral tests we did, they said they were cognitively normal, but their brain's functioning differently.
Yeah, this is deep stuff.
Yeah.
Well, yes, we're probably running out of time, and we've really covered a lot here. We may want to talk to you again. We're hopefully going to be working with a group in this field to look at like harmonizing omega-3 levels if we want to have a standard level that would identify a low intake or a low omega-3 status. Having a a more harmonized group or decision on that target, because it is a little bit all over the place. So when you're looking at research in this field, there's a lot of caveats. And so I think that was really kind of a big part of this year's ISFAL conference, and it's something that has been on people's minds for a very long time. But I think what's happening in Australia is really kind of pushing this forward. And then your study coming out also is saying if this is going to be used clinically, then we should, on the research side, we should at least standardize so we can tell the clinicians how to do this. So hopefully that'll be something we can update a little bit later. Anything else that you want to say about the study and about what's coming up next?
Well, I would just add to what you said there, that I have a colleague who's doing studies in this area who did not realize until about a month ago that when you look at the DHA level in Olson's study or Sheila Innes's study, it's not the same. So people are doing these studies and they read a number and they don't say, well, oh, it was plasma, or it was red blood cell phosphorus, or it was blood spot, they just look at the number, and that was actually one of the reviewers of this paper had read Olson's paper on plasma and said, well,
yeah,
they're 4%. So why are you using 6%? Well, then I had to explain that plasma has very low levels of these, the DHA compared to a red blood cell phospholipid, for example. So it is confusing, and it's not practical now for obstetricians. So I do think that we need some harmonization. The problem here is we need to get it out there with like a little table that everybody can go to and refer to quickly. Is it a plasma measure? Is it a blood spot? Is it a red blood cell? What is it?
And moving forward, having a more coordinated effort to make sure we report like the same things as much as we can, and what fatty acids. So that's a huge area.
Yeah, the one thing that I would say is that I've been encouraged by obstetricians' responses to this data that are picking it up, and the obstetricians, there were 4 on this study, and they were kind of blown away by this because they really don't have any way to fix these. There's actually some data out there that you can predict who's going to have an early preterm birth, but they don't have any strategies to prevent it except cerclage and progesterone, which don't really work after a certain time. And this trial was interestingly done where women who'd had a prior preterm birth before 34 weeks actually got all the treatment that is recommended by ACOG. So it's a real world trial, and we still reduced it from 4% to 2%. The women who'd had a baby in this study before, 7% of them had an early preterm birth. So it was really high.
Oh.
And the fact that overall we got a 2% rate in the studies tells you that something was working.
Yeah, very
some
interesting.
of their interventions, and as well as DHA as well.
So are you getting champions among the obstetricians that you know who would maybe push this kind of thing at ACOG?
Well, at this point, I'm kind of waiting to get this paper out that Sarah's doing with the red cap survey.
Yeah.
That could be implemented anywhere.
Right.
And, one of the obstetricians here just embraced it instantly, and he actually wrote the red cap survey.
Oh, great.
So I'm encouraged, but I don't want to push it too far because I think it, what I asked the obstetricians on the trial to do was put this up on your Facebook, your LinkedIn, because obstetrician, I was a professor of OB-GYN once, but I'm not currently, and they're not going to listen to me, but they will listen to their peers, right?
Right.
If their peers are getting excited about it, I think
that's good. It's exciting. Exciting.
Yeah.
Thanks a lot, Susan. That's great. Really appreciate it.
All right. Talk to you all later.
Okay.
All right.
Thank you.
Bye-bye.